the most challenging thing with sickle cell is understanding that, you know, some days I'm gonna have up days and some days I'm gonna have down. This is your body. This is what you use every day. Having sickle cell, you're not allowed to, I feel like, enjoy some of the little things that other people enjoy. I feel like obviously we've been dealt a crappy hand of cards, and you know, the goal is to play it to the best capabilities that you can.
Sickle cell anemia is a terrible disorder. It is a hereditary disorder. That is, it's something that occurs in one's gene, inherited from parents. Yeah, worldwide, it's the most common hereditary disease. So many millions worldwide. In Africa alone, 250,000 newborns are affected annually. So the hallmark of sickle cell disease is pain. It happens when, after the hemoglobin molecule, which is specifically designed to bring oxygen to our vital organs, delivers the oxygen, the sickle mutation, which is the change in the DNA, creates this long strand, making the round, flexible red blood cell into a very sticky, stiff, sickle-shaped, crescent-shaped red blood cell. And that blocks blood flow. So when you don't get enough oxygen to a tissue, causes pain. So patients who have sickle cell disease experience pain. They experience damage to vital organs where blood flow can be altered by the sickle red blood cells. Lung problems, when you don't get enough blood to your brain, causes a stroke. These are all common complications that affect the quality of life, not just the lifespan in a person with sickle cell disease.
I feel like I've experienced some, you know, really crazy amounts of pain. Like I've had both my lower extremities, you know, they had infarctions in them. So, you know, like imagine both your legs just being broken at the same time. It was the worst pain you can think of. And then my, like with my chest, my pericarditis, like I could barely breathe. I was in like constant pain all the time. So there was some days I just felt like a zombie, you know?
We know that if you correct that mutation, you take away the disease. So anyone could imagine that if it was possible to correct the gene in one's own cells, that you should be able to cure the disease. So when we first heard about CRISPR-Cas9, we got very excited because that could target the sickle gene, correct the problem, and then restore healthy red blood cells in the circulation. There are 3 groups of scientists and clinical investigators working together to accelerate the development of this technology in humans with sickle cell disease. So one group at UCLA will be involved with manufacturing, that is, producing the blood stem cells after correcting the sickle gene, and they are expert in that activity. The group here in Oakland are experts in hematology and sickle cell disease, and ultimately will lead a clinical trial when we begin to treat patients. Yeah. And then the anchor, I would say, is the IGI, the Innovative Genomics Institute at UC Berkeley, that really brought that technology to us. And they're leading the effort in documenting, at the DNA level, how many of these blood stem cells we've corrected. We don't have enough therapies for sickle cell disease. If it's related to societal pressures in underprivileged, underrepresented populations in the United States, how rewarding it would be to deliver this amazing cure to this underserved population. It's justice, right? It's the first hereditary disease ever defined in human beings, primarily affects African Americans, an underserved population, has difficult access to healthcare in some cases, and now finally, we have a cure that we can offer the majority of patients, at least in our country. I mean, that would be terrific.
I think the thing that's amazing about science and about biology for me is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.
There are the sick, there are the hungry. The planet is growing, the planet is warming. We need to do things about that, and we can.
We have the power to change things using gene editing.
CRISPR, in principle, is the solution.
What CRISPR does is provide scientists with a tool to change DNA sequences in plants, animals, humans.
CRISPR has the possibility to end disease like sickle cell anemia, HIV, malaria.
To build crops to meet the needs of a growing planet.
The potential is just incredible.
It is very important for people to understand the technology CRISPR.
The mission of the Innovative Genomics Institute is to take the marvel that is CRISPR and the extraordinary power of precise genetic engineering to make a difference in the real world. How does CRISPR work? Imagine you're trying to edit text. What's step one? You have to identify which section of text you want to edit. CRISPR can be programmed to find a specific stretch of DNA inside a living cell. It runs inside the cell, goes into the nucleus, and finds precisely the gene that we asked it to find. And then it's very much like word processing. You hit backspace, backspace, and you type in new text. What is the molecular equivalent of backspace, backspace? Believe it or not, you actually cut the DNA. Cracking the DNA open gives scientists the opportunity to change DNA precisely where the break occurs. So, for example, we can get rid of a gene that causes a plant to be susceptible to a disease, or we can add a mutation that will give a plant a beneficial trait. We are very mindful of the fact that we're not building solutions in a vacuum. We're building solutions that people worldwide will want to accept and use. And yet, history teaches us that if we just show up and tell the world, look at our CRISPR-based solutions, better animals, better humans, come all, take a ticket, get in line, that's not what's going to happen. Some innovation is welcome, other innovation is not.
Whenever there's a new piece of technology that's being developed, for example, CRISPR, most people have the first reaction, fear. It's scary. What is going to happen with gene editing, with this new powerful CRISPR technology? But at the same time, how are we going to take advantage of this very powerful piece of new technological invention without the fear?
10,000 years ago in Neolithic China, folks domesticated a wild plant which we now call rice. In process of selective breeding to give themselves a grain that feeds them, The Neolithic Chinese introduced a wealth of genetic changes into the DNA of that crop. And here we are with many varieties of tasty rice, not one of them genetically looking like the original parent. The 20th century has been the first one where humans have started to take directed control over their own genetic destiny and the genetic destiny of the world around them. CRISPR is different because of our ability to not engineer Mother Nature, but instead to collaborate with her.
This is our machine that we use to deliver DNA to tissue. We call it the gene gun. That goes in. Then we put our DNA in, and finally our rice cells. We create a vacuum in the chamber and we allow the pressure to build up, and then at a point it breaks and it shoots the DNA into the tissue. So these cells now have new DNA inside of them. Over the next few days to a week, the DNA will start to make its way to the nucleus and we'll start to make the edits that we wanted of them.
I got it. Thank you. So this is the tissue culture room. Here we grow our plant materials. So Jackie just introduced the DNA and CRISPR reagents into the plant cells for us. Thank you. And now we need to put them into the dark because after the stressful treatment, they need to have some rest. And also we need the CRISPR reagent to be active and actually make the cut. So hopefully in the end, we will have the ideal rice plant. And for example, this means we increase the yield, the grain number a lot through adding more branches to the flower. And this really has a high impact because you get more grain for one flower. Then we will also reduce the number of stems in the plant to make the plant more compact. And this has a super high effect because you can grow much more plants in a smaller field. So you get also a much higher yield. Honestly, if you ask me, I think it's a revolutionary technology. Already at the IGI, we're doing 15 different plant species that we try to improve. We work together with agricultural institutes around the world, in Africa, in Colombia, and in Mexico. They're going to help us to choose the best plants. So we really think we're going to have a high impact in different environments that are maybe troubled by climate change, or even in environments where the soil is poor.
In cancer therapy, we know that cancers are driven by mutant genes that drive cancer cells to grow. In this case, we know that cervical cancer is driven by a virus, and this is a unique gene that drives the cancer. We've used CRISPR to target that gene, and that kills only the cancer cells, leaving normal cells untouched.
Basically, we cured cancer in animal models entirely. So cancer cells or the tumors have been cleared 100% with 100% survival rates for every animal used in this study.
So what we did is we took human cancer cells and used them in our preclinical animal models. We gave them cancer.
Then we refined and designed a specific treatment that targets certain genes within the DNA of that cancer cells.
What CRISPR does is it targets the gene and it breaks it open, and the body tries to repair it by adding a few extra letters. It's like misspelling a word, and the spell checker no longer recognises that as a right word. The gene doesn't make any protein. The cell dies. So to our knowledge, this is the first example in the world of completely curing a cancer using CRISPR technology. Others have shown reductions, but this is the first complete cure. We're really excited.
This study from Griffith University is the first to demonstrate successful treatment of metastatic cervical cancer using CRISPR gene editing. Our aim is to translate this exciting finding into the practical treatment for our women. To help achieve this, we have weekly meetings with these exceptional research scientists aiming to work out ways this can be done.
We are hoping to see our treatment being applied to human to treat cervical cancer and other cancers, hopefully in the next 5 years.
One of the main things to determine there is to make sure it's safe and effective and decide how many treatments we need to give a patient before they're cured of their cancer.
If we are able to identify the driver genes that drive the cancer cells, regardless of the type of the cancer, this means this technology will be able to be utilized to treat that kind of cancer.
There are labs all over the world who are developing CRISPR technology for cancers. And CRISPR technology is really the next big thing in terms of gene therapy. Things which have a very poor survival rate now. This technology is going to revolutionise how we treat cancer, and survival rates will improve dramatically in the next 10 years.
Jennifer Doudna, ladies and gentlemen. My name is Barbara Osher. I'm the Honorary Consul General of Sweden in San Francisco. I would like to welcome you to this very special occasion in Berkeley. Since 1901, the Nobel Prizes have been presented to Nobel laureates at ceremonies in Stockholm and Oslo on the 10th of December, the anniversary of Alfred Nobel's death. In his last will, Alfred Nobel expressed that he wanted to establish a prize and award those who have conferred the greatest benefit to humankind within the fields of science, literature, and peace. Jennifer Doudna, the Li Ka-shing Chancellor's Chair in Biomedical and Health Sciences at UC Berkeley, as announced in October, the Royal Swedish Academy of Sciences has decided to award you the 2020 Nobel Prize in Chemistry to you for the development of a method for genome editing. Together with your fellow Nobel laureates, you will be celebrated on December 10th in Stockholm at a prize ceremony in the City Hall, attended by members of the Nobel Foundation and the Nobel Prize awarding institutions. On this occasion, the work you have been awarded for will be presented in more detail. And customarily, you would have received this prize from the hands of His Majesty the King. However, this year you will be celebrated with the distance forced upon us with this pandemic. And since you cannot go to Stockholm now, your Nobel Prize and medal and diploma has been brought to you here instead. So, with regards from the Royal Swedish Academy of Sciences and the Nobel Foundation, it is a great honor to convey to you my warmest congratulations and ask you to receive the Nobel Prize.